Hello, and welcome to Open Science, Open Data, Resources and Methods of the Kidney Precision Medicine Project. The drive toward precision medicine in nephrology was the impetus for the Kidney Precision Medicine Project. Open source data and tools are key elements of the FAIR principles to engage a wide spectrum of communities, democratize scientific efforts, and increase transparency in research. They inspired the development of various analytic methods to highlight different aspects of public data and are benchmarks for validating emerging computational techniques. The Kidney Tissue Atlas is the data sharing and visualization tool developed by KPMP. The aims of the project were to create a comprehensive multimodal resource, design it for broad public access, and then encourage application by facilitating data searches via user-friendly web tools. The project also firmly ensures participant data privacy while fostering open science. It is the hope that this resource will eventually contribute to drug discovery and development. The KPMP knowledge environment integrates diverse types of data, including clinical, histology, regional data, bulk sequencing data, cell-level sequencing data, and spatially resolved molecular profiling data to form the Kidney Tissue Atlas. The Kidney Tissue Atlas is comprised of 4 elements: a biorepository of tissue samples from which data is generated, a data repository from which users can download raw and processed data, an explorer that highlights cell markers and summary data visualizations, and a spatial viewer to explore spatial data. KPMP data is made available in a variety of formats, including H5AD files compatible with established programs such as Seurat or Cell by Gene. This allows the user to perform advanced visualizations, verify findings, and also to use existing data as a building block for new analyses. A data analyst, for example, can access the Atlas to download raw data and analyze in their own environment, running it through the suggested pipeline or modified one. In this way, pipelines and parameters can be assessed for adequacy, and results can be evaluated for accuracy and reproducibility. A data analyst can also download the Seurat object, change the parameters, and rerun the analysis, or even combine it with other data to serve as a starting point for other analyses. In another example, a clinician or biologist can investigate a specific research question, such as where in the kidney do SGLT2 inhibitor effects take place. Inhibition of SGLT2, a sodium glucose cotransporter encoded by the gene SLC5A2, has shown great promise in treating kidney disease. The cellular location and expression levels can help uncover the mechanisms involved in its therapeutic effects to better tailor patient treatment. Typing the gene symbol into the Atlas Explorer generates a visual display of kidney cell types, expressing SLC5A2 and specificity of this expression in each cell type relative to all others, as you see on the left. Further, dot plots enable comparison across regions and disease subtypes. As you can see in the figure, SLC5A2 has the highest expression in proximal tubule cells in the healthy controls, less in acute kidney injury, and very little in chronic kidney disease. This suggests disease-specific variations in expression. The associated cell by gene feature within the atlas enables visualization of gene expression at the cell level in biosamples of interest. Dots represent individual cells. Despite a higher number of proximal tubule cells from CKD compared to AKI, the more pronounced loss of expression in CKD versus AKI relative to normal is more obvious, as visualized here in the figure. The KPMP provides both tools and a multimodal repository of kidney-relevant data. The web tools allow for an interactive exploration of the processed data. Additionally, both processed and raw data can be downloaded and imported into other applications or used for other analyses. KPMP also collects phenotypic identifiable protected data that is available with approved requests. The data in the Kidney Tissue Atlas is carefully curated by experts in the field. Additional data is generated and added to the repository as protocols and processes are established, optimized, and shared. KPMP is a dynamic project that welcomes user interaction and engagement in the community through feedback and suggestions. The repository and content are shaped by the project, data generation requests, and ancillary studies. The Kidney Precision Medicine Project seeks to ensure open science is available and achievable to all stakeholders, including investigators, clinicians, researchers, pathologists, students, and the participants themselves. Thank you.